All right. In 2004, our objective was to breed productive, gentle honeybees with tolerance or resistance to mites and brood diseases. Now, 20 years later, I'll give you an idea of where we're at. I think everyone would agree that the current honeybee health issues are mainly from Varroa, the viruses it transmits to the bees, the miticides we use to control it, pesticides in the environment, nutrition, both in the environment and the compromising functions of the Varroa, where they suck the fat on the fat body in adult bees, and hemolymph during reproduction in the pupa. So just to briefly go over what we're going to talk about today, mainly the Saskatraz, the breeding and selection program, and a focus on some of the selections we use for economic traits, honey production, and so on. Selection for Varroa resistance and hygiene we will look at in more detail than usual. Natural selection, VSH phenotyping, and the UBO assay in particular, and what we've done for screening for grooming behavior, mite biting, virus, and nosema susceptibility. But I won't be talking about nosema anymore because we've quit selecting for it. We just don't treat for it. Biomarker development, the main focus today will be on micro and genome arrays. We have done a lot of experiments on combined miticide treatments with selected and non-selected stock, and really the bottom line is that if you use selected stock, you get better efficacy with your miticide treatments. And lastly, the Saskatraz hybrid project I'll briefly discuss is how we distribute the genetics we've been selecting for. So our primary selection criteria has always been honey production, wintering ability, and spring population growth. These traits go hand in hand. If there's good population growth and wintering, there's usually good honey production associated with the colony. Varroa resistance and suppression with some new techniques that we'll look at today. Resistance to brood diseases has always been a selection criteria, and virus susceptibility we'll be talking about somewhat. And we'll discuss the breeding methods that we use to enrich for these traits. For Varroa resistance, we use natural selection. To maintain the lines, we outcross, backcross, and recurrent selection, progeny analysis, and closed population mating are all techniques we use. The breeding program logistics is in this slide. It looks rather daunting, but it's quite simple, really. And the history, we started at Meadow Ridge in 1992, selecting for good winter survival, honey production, and resistance to brood diseases, in particular chalk brood. But in the early 2000s, we ended up getting tracheal, or even before that, tracheal and then Varroa infestations, and we had no resistance in our population. So we introduced German and Russian selections early, and we also had Canadian selections introduced by Saskatchewan and Manitoba beekeepers in 2004. So we got a lot of diversity, you'll see, putting into this natural selection apiary, which we started in 2004. We added the VSH trait in 2012 to this, and then we continually select every year. So what we do, introduce the selections into the natural selection apiary, select the best honey producers and Varroa resistant colonies, and select for survival. We outcross the best and then backcross to the same apiary, which we did to try and enrich for the traits with the drones that were subjected to Varroa population growth. So in any event, the bottom line is here is that recurrent selection is the key word. We keep doing trait selections, and we've distributed stock since 2006, at least in Saskatchewan. So just to flesh this out a little bit, We use a lot of close population mating, have used instrumental insemination, but it's easier for us to try using close population mating. So in progeny analysis, we try to stabilize or balance phenotypes by crossing our best Varroa tolerant or resistant lines to our best honey producers. And that's done under close population mating. Natural selection, we still run that and put colonies in continually, reselect them to naturally select for resistant traits. Economic trait selection is done annually from about 1,500 colonies, selecting for honey production and winter survival. And we label all of our colonies. This is an example. We have the family, we can identify the mother's ID. Sampling ID is a short one, and the year mated and the location of the colony. And the unique ID contains all the information that we collect for that colony. So every year we select all of our apiaries. We look at best survivors, colony strength, food stores, clean bottom board, so on. Mite washes are done on the best colonies and the weakest, and we get a composite yard sample. Honey production, we mark the 2 top producers in every yard at each harvest. Tom does most of this. Final selections are made on or after the colony's 2nd year, and these are moved to an isolated area for apiary for close population mating, and they are reselected for the best breeders and drone mothers. So here's an example of the apiary we've been using for a number of years. You'll see on the right y-axis the honey production in pounds, and on the x-axis the different colonies that were selected and put into this apiary. And just to point out a few things, this one in particular is of interest. It was an S28 colony that was put in first in 2012. The colony tends to supersede quite often, but you'll see in 2020, even though it produced 150 pounds after superseding, it's starting to regain its production. S14 is a colony that shows quite consistent honey production over the 3-year assay. S96 is similar, and you can pick out, sometimes we put in a colony with excellent honey production that we graph from to do progeny analysis on it. So the next series of slides, I'm going to look at our natural selection apiaries for Varroa tolerance, and this one is in 2018 study, and we do this every year. We measure the phoretic mites, we measure the honey production, and you'll see that we score the mites in the spring, the green bars, and in September is the blue, so we can follow the growth, and the honey mean is the yellow line. Colony mean, mice per 100 bees, is the red dot, and yard mean phoretic infestation is the red bar. So generally, in terms of selecting, we look at colonies that show good honey production, so above the mean, and show good suppression of Varroa mite growth. So S26 stands out here. So does G20. So we would be multiplying those. But the SY26 I want you to pay particular attention to. This is a phenotype that we're looking for. And you'll note it's been in the yard since 2014, still surviving. It's superseded several times, but that colony is consistent at keeping the Varroa levels low. But we multiply that, of course, and this is the same chart, same coordinates, but we measure the mite levels in May and July and August here. But if we track the daughters or granddaughters of the SY26 that we noted in the previous slide, we see that there's considerable variability within the daughters. So that's an issue that we have, is stabilizing this Varroa resistant trait. Just to expand a bit on that, we look at a few of the daughters here. You see same coordinates again. We see that the SY26 116 is quite good at maintaining low varroa values. So in the spring, the green bar, it shows some infestation, but it's even lower in August. So that's a good sign. And we have a few others. SY2123 was pretty decent for keeping varroa low, but it shows a bit of increase in the fall. Another colony that's of interest, and we'll talk more about this later, is S96 that shows maintains low varroa populations. And before I expand on that a bit, I'm going to talk about progeny analysis here again. In this particular analysis, we're looking at S113 breeder, the phoretic mite levels on the y-axis on the left, and a number of the daughters on the x-axis, and we measure the colony mite levels in November and again in April. And you'll notice that in 3 of the daughters, those varroa populations have dropped to 0 or non-detectable. So this tells us that this colony is able to reduce the varroa population over winter, and that could be by grooming, mite biting, and also VSH activity, but it's something you can all do fairly easily. Well, just an example of a grooming video that I do. I do this by trying to get varroa onto the bee, and you'll see this Bucky the bee is vigorously trying to remove it by auto grooming. However, none of her sisters seem to notice that mite. It's interesting that there's no allogrooming in this case, even though they crawl right over her. It's just that they cannot see it, and they cannot smell it either for some reason. I think it's because the varroa mites tend to be able to mimic the cuticular pheromones of the bees and do it quite readily. Within a few hours they've achieved it. The next video shows mite biting. I placed a pupa with a varroa on in front of them and watch their reaction there. This particular bee grabs it and bites it with her mandibles. So you can see this happen on occasion, but more often than not, they don't notice the mites because of the mimicry that the varroa has developed. Parasite survival. This is an example of some work we did a long time ago. We don't spend too much time on it anymore, but it clearly shows that if you evaluate by microscope analysis the varroa on sticky boards, you can end up scoring the number of mutilated mites. Greg Hunt is in his laboratory, and now there's Crispin Given has given some information on this. They have found that Nerexin transcripts are correlated with this activity, which they can measure, which is quite nice. This is the work that we did, and we just looked at the percent damaged varroa in a number of different colonies at Saskatraz Natural Selection Yard, and found that 3 showed quite a bit better damage, but you'll notice the percent damage is just a little over 17 to 18%. But those colonies survived longer, and 24 and 6 rapidly were destroyed by varroa. The next series of slides I'm going to show you is what we do to look at how our phenotypes handle viruses. On the y-axis you see the CT value of deformed wing virus detection, and on the x-axis the colonies that we tested. But I'm just going to point out a few. So basically what we did here was isolate the head tissue from these SY26 strain and S23A at that time, and measured by qRT-PCR the level of viruses, deformed wing virus, in bees that were infected with mites and ones that were not, without mites. The blue is without and the red is with, and you'll see that in these two the infected and uninfected show about the same level of deformed wing virus infection. However, if you look at a Varroa susceptible line, you'll note that the without mites is much lower than the with mites. And remember that CT values, the higher the value, the lower the virus titer. In the next slide, we show the same thing on the left panel. It's just a different way of presenting it. And the susceptible has a much higher level of deformed wing virus. But something that is of interest and should be pursued is that on the right panel we see the effects of treating, not treated, control, apivar treated, apistan treated, and thymovar treated, and we expose the pupa to these chemicals, and we see in the SY26, the tolerant line, there's not much significant difference in the level of deformed virus present in the pupa heads. But if you look at S14, it's similar. It's also got some good resistance. But the susceptible, and particularly in the apistan and apivar treatments, have quite a higher level of deformed wing virus. Thymol had the least effect. So something of interest, we may be weakening our colonies by treating to these pathogens. I'm going to switch gears now and look at the biomarker development. We've done a lot of different things, microsatellites and proteomics, but today I'm focusing on microarrays and our transcriptional expression and on signal transduction with genome arrays. So first of all, this is some of the transcriptional work we did, and what we used here were two extreme phenotypes, a colony G4, which was very susceptible to Varroa. The Varroa just ate it up, and SA88 would survive 58 months with no treatment. And this was done by two graduate students, Sanjay Chang and Cherry Wan, over a period of several years. This slide is just a small snapshot of what we did, showing some significant changes in transcripts. In the tolerant colony, we've seen quite a large increase in apolipoprotein D-like transcripts, 7-fold increase in this ratio, but in the actual case with the tolerant itself and the G4 minus, it was something like 14-fold. So dramatic increase. Esterase E4-like transcripts, there was 3 to 4-fold increase. That particular component or esterases are involved in detoxification mechanisms for pesticides and likely miticides as well. Cytochrome P450 oxidase also involved in detoxification mechanisms. There's many others that I don't have time to talk about, but also of interest. This is an analysis that we were the first to do, and to me it's very, very good. It's first responders, the genome kinase activity, and in this panel on the top you see an array of 299 peptides. Each of these peptides are 15 mer with a phosphorylation site in the middle, and when they're phosphorylated they light up. If they're dephosphorylated they don't, and the level of phosphorylation can be monitored by analyzing the fluorescence intensity. One thing that we were really pleased with is that we could see consistent and repeatable displays. So in other words, the replications were consistent. So we were pleased with that the experiments were mean something. So we subjected the same phenotypes, G4, infected and unaffected, and S88, infected and uninfected, to analyses. And to make a long story short, we identified 19 phosphopeptides which could discriminate between the resistant and tolerant lines, and most of them were involved in innate immunity and stress responses, which we've also correlated with our transcriptional analysis. So we subjected this to principal component analysis to try and separate out the phenotypes, which we did clearly here. The G4 is the green dots, and the square dots are uninfected and the round infected. And we see a separation on the basis of infection status. And that separation was, by bioinformatic analysis, the results were that in the susceptible colonies, Varroa infestation was lowering or down-regulating the innate immune factors. So the bees were more susceptible to virus infection. Whereas in the tolerant line, S88, we see in the red dots and squares that these are not affected as much, either infected or uninfected, but there was increased upregulation of innate immune factors in the tolerant, and there was no effect of the Varroa on downregulation of these. So that's an important mechanism, I think, for survival of honeybees, is a susceptibility to viruses or pathogens. So we should screen just ordinary colonies for that and look for correlations. But I'm going to talk about in more detail about hygienic behavior traits. Of course, spring evaluations I mentioned before, clean bottom wards. Freeze kill tests we have done in the past, but we no longer do it. Really, it's not that well correlated with Varroa resistance, and the bees are just detecting the death pheromone, or oleic acid in this case. Host brood suppression is of interest in suppressing Varroa reproduction. There's some recent papers on that. But the bees that they work with are not economic and would not be of interest here. So we use natural selection, and this takes a while before the Varroa populations build enough to do some proper assessments. We also have looked at uncapping and recapping behavior. And we always, if you see a lot of pupa with Varroa mites on them laying on the doorstep of the hive, that's a sign of a good removal behavior. We monitor the Varroa populations, honey production, and viruses when possible. So we look to select the best over time, survivors. We have done Harbo assays in the past, but have done some more just recently in combination with the UBO assay. So it's a single colony assay, which takes us about 45 minutes. We score about 200 brood cells at about 7 to 11 days post-capping, which represents the purple-eyed or tan joints to purple-eyed gray wings for non-reproductive mites. It's important to emphasize here that the bees that are VSHBs remove reproductive mites, which means there's mite families in the brood cells that are non-reproductive. UBO assay is an unhealthy brood odor this was developed by Kyer Wagner and colleagues at University of North Carolina. And we have been able to do 50 colonies in about 4 hours, so it's much faster. This assay is based on cuticular hydrocarbon pheromones that are given off in bees that are infected with a Varroa mite or a family of Varroa mites, or it works with viruses too, apparently. So essentially, it's a mixture of 4 synthetic monoalkenes that are synthesized based on what they've detected in the brood. They're diluted in 1/2 ml of hexane solution and sprayed on 50 cells. So then they're placed back in the colony and scored after 2 hours. And it's a rapid way to select for a VSH activity. Just an example here, some of the first we did of the unhealthy brood odor assay developed by Kyer Wagner. So the analysis here was 84.4% assay, which is quite good. So this colony should have good VSH activity and keep Varroa mite levels down and give better survival and so on. So in the next series, I'll show you some videos of what we actually do in the yard. In this case, Antonio is removing a frame that was marked the day before. So we don't use any smoke during this assay and try to have minimal disturbance. The comb is removed and pictures taken of the notes on the frame, so we keep track of the colony and its origin, and the brood we photograph, so we have a record of what the brood looks like. And then Colton checks on the stage of the pupa. They should be at the purple-eyed stage, as previously noted. Then we photograph the ring again to make sure we have all the data we need, because it's assessed in 2 hours. So, and this is the application is 3 short sprays to deliver 1/2 ml of the hexane solution containing the hydrocarbons mentioned. There's 2 longer chain and 2 shorter chain, and the longer chain are supposed to allow the bees to pinpoint, whereas the more volatile shorter chain have the sort of a general alarm where the specialized workers are attracted to. So now that that's complete, it's put back in the colony, and in 2 hours we assess the results. This is how not to do the assay. This was my first attempt at it, and I holding it too close and not spraying too fast, too trigger happy. So I'm given direction here by the expert Colton, who has done 174 assays last year. So note that you should wait a little while between sprays and try to keep the distance proper. With practice, you get better, and just try and not miss the frame complete, or there like I did there. That's not a good application, but anyhow. This is some assays we did previously when we first started on some Australian colonies, and before and 2 hours after spraying these compounds, there's no response. They are very naive to it, but have not, as of yet, been subjected to selection pressure or to varroa. So they have not evolved any scent of varroa or resistance mechanisms. So they have varroa now, but it'll take some time. This is some of our own selections from a particular line that we've been working with for a number of years, SY26, which has shown very good, very good resistance in the past. And just note the SY26-123, which showed a good score, over 64% UV assay score, and decent honey production. So that one was a candidate for us to multiply, and we'll show you the progeny analysis on that later. This is an example of the UBO scores over the natural selection apiary from fall of 2022 to the fall of 2023. There was 6 assays done on each colony, and there's something to note here that the colonies that show a low score show the low score pretty consistently. Now this is a box plot which shows the distribution of scores over the 6 replicates. And some of the colonies that score higher tend to have a wider variation. For example, 124 has quite a variation in score from lower to higher. And 123, which we'll talk about later again, shows that close to 70%, and at certain times we'll see they're lower. So there's seasonal variation is what we found. So here's some more detailed data. On the left panel we have phoretic mite levels, and on the right-hand panel the UBO scores presented as percent. And we see that in the S113 that has a low UBO score on all replicates, that the mites tend to start to grow quickly in August. So this has a low UBO score and likely low VSH activity. If you look at S96, we see that it has virtually no response to the UBO, but it does keep the mites down. There's some increase, but not as high as the previous colony in the fall. SY26116 shows a very low UBO response, and it keeps the varroa population right down. So this colony is displaying resistance, but not a high UBO score. In the SY26123, we see 3 replicates that show good UBO scores, but then in July, the score goes to non-responsive, but we found that this colony actually superseded during that time. So that may have an effect on the UBO response, but it did keep the varroa down, and then it slowly starts to creep up in September. The SY124 showed a very good UBO response in the fall of 2022, but it was decreased response in the summer months, and then in the fall it did show some, but notice the varroa population just skyrocketed starting in July. So this one didn't correlate well with what we would expect to see. This time we're looking more at the Harbo VSH scores in correlation with UBO. We see the percent brood infestation as the blue bars, percent non-reproductive mites, the orange, and phoretic mites, the red dot. Notice in the S113 that it shows a decent Harbo score in the spring and June, but the mites rapidly increase, and it has a low UBO score, which is consistent with what you would think. The next panel shows S96, which is showing a good VSH phenotype of 4, but a low UBO score of 2.3 maximum in the spring. But then in the fall, it's the varroa levels start to increase, and the Harbo score goes to 0. So this colony may be under stress from heavy varroa infestation in the yard, but if surprisingly in the fall it went in looking quite good with no expression of any form of deformed wings present visibly. So an interesting phenotype. The next colony is also interesting in the fact that it had a low UBO score, but it kept the varroa levels right down and an intermediate VSH score. Then again, the colony that showed the highest UBO in the fall of 2022 shows some varroa increasing in June, but a very quick increase in varroa levels in the fall, and very low Harbo scores, so not a good correlation with the UBA in this particular case. I just want to point out that SY26123 showed a good UBO score, a reasonable Harbo score, and low varroa population growth, but it did supersede. This is the colony we selected for progeny analysis, and we'll show you some of those results here. So this is these are I think 53 daughters of SY26, the maternal line, and the UBO scores on the y-axis. And we're looking at a box plot again with the spread of the UBO scores for different mating apiaries. So Bainesville shows the highest mean value, and Poncelet the lowest, and Purdy's the second lowest. But the main point here is that we note that the Bainesville yard, which is natural selection yard, has given us the highest UBO scores, and that's because the paternal lines there likely carry more of the VSH traits that we're looking for. So the take-home message here is that the patri lines are of importance. We also noticed that in our analyses, the correlation with the phoretic and brood infestation was of interest. On the y-axis is the phoretic mite infestation, and the x is the brood. If you take a look at 15% infestation of, or 20 of phoretics, you're going to have a brood infestation of about 40. But kind of useful information. So just to summarize what we've done here, we've used the UBO assay to try and predict VSH phenotypes. We have some, in our experience, some considerable variability in the response over the season. Some colonies that do not respond show low overall population growth, so we're interested in multiplying those too, but there may be different mechanisms involved in this. So we're selecting the highest scoring colonies to establish the close population mating area, apiary, and then we'll be back crossing to that apiary the best UBO scoring maternal line, and also RS96 and SY 26116. And then once we produce that progeny, we'll be going through the same process again. So just to summarize what we've been talking about, when you select Varroa resistant colonies by natural selection, you find survivor colonies, and what analyses showed us. We had two colonies that were exceptional, S88 for 58 months and SY26 for 70. And what we found is both of those colonies showed very good VSH phenotypes. We also found that some of them were very good groomers, some mite biters, but I believe that grooming is not the major factor in Varroa resistance. I think the mite biters, though, are important because they tend to have, I believe, traits involved with detecting Varroa, better bloodhounds, so to speak. And we noticed that supersedure, brood breaks, and requeening helps with controlling Varroa population growth. So nuclear production is a good way to keep Varroa levels down. And with our biomarker analysis, particularly the transcriptional analysis, showed that certain transcripts are increased in quite significantly in resistant colonies. Apolipoprotein D, which is an important protein in the hemolymph of bees, which is involved in immune responses and blood clotting. Esterase E4, involved in detoxification of pesticides, miticides, and other possible factors. Cytochrome P450 is also involved in detoxification mechanisms. In the genome work, very important discovery that in the most resistant and survivor colonies, that the factors involved in innate immunity were upregulated and quite significantly, where the Varroa managed to downregulate those factors in susceptible colonies. So an important thing that we shouldn't forget about in terms of selecting. We've noticed and just done some work with the USDA. Katie Eiley has done some work on vitiligin and different strains, and the Saskatraz strains tend to maintain vitiligin transcripts even though there is dearths in pollen. This may have some effect on wintering ability and, of course, survival. And most of these survivor colonies are better foragers and better, therefore, likely better nutrition for the colony. So to summarize all this, our goal is to combine and stabilize the traits associated with the resistance mechanisms we've discovered, and to do that by recurrent selection and close population mating. So our strategy is to work with the colonies and the strains that we already have economically selected, and try and find the most Varroa resistant or tolerant colonies within that strain, and to screen them by natural selection, UBO assay and Harbo assays, and improve the heritability, which is our biggest problem, is stabilizing these traits by combining the best paternal and maternal lines. And there's been good work done by the Hilo project and others on maintaining that using instrumental insemination. So we hope to keep using our genome assay, which we have developed further now that should be useful in selecting just colonies at large, and to further understand mechanisms involved in resistance and discovering some more informative markers. So once we have these traits, how do we pass that on to our other beekeepers? Well, in about 2012 or so, we initiated the Saskatraz hybrid project, and the reason was that we'd had some requests from commercial beekeepers in Canada that wanted to obtain larger volumes of our stock earlier in the year, in April. So the way to do that was to establish a queen production program in California, although we tried other places. This worked out the best. So our stock that's selected, as I've described, for honey production and wintering and so on, were selected during the production year in Canada, and we shipped breeders down to California for reproduction. I'll talk more about that in a few minutes, but essentially this project was designed to supply larger volumes of hybrid queens with selected in Canada with the genetics from that, and this would result in increasing the frequency of alleles associated with economic traits and with varroa resistance in commercial populations. And the reason for that, of course, is that the drones are copies of their mothers and would carry those alleles and integrate them into the population. We've distributed stock to a number of countries, which you can see, and there seems to be continued interest, so I think they're performing well in most places. On the right-hand side, you'll see David Powell in the top right panel, who I initially started with, and in the bottom, you'll see me and Josh Oliveris, Ray's son, working some bees, some selected breeders in California. This is some of Ray's crew here. On the left is Josh, Ray, and one of his assistants, Gomez. And on the middle picture are the ladies that do most of the grafting and looking off after the cell builders, and Jose Gomez again with Dario, and they do a real good job and run a high number of cell builders, up to 900, I believe. So when we look at the locations in Canada, we're lucky if we can raise queens for 3 months of the year, whereas in Orland they can do it up to 6 months. So we have a restricted time period, and that's because the temperature range is very wide in Canada. We've had up to -50 or better, whereas in California it's rather longer, nicer season. So just to review what our objectives are here, we produce queens in Saskatchewan, breeder queens focused on the methods I've described: recurrent selection, close population mating. Just short seasons, so we can only produce not as many as we'd like, but we can end up getting around 2,500 or so, and we pre-select about 150 or 200 of those to send to California. So there they are a large-scale producer of queens, and we reselect about 40 to 80 of the best, and their climate is ideal for starting early. They start grafting in February, end of February. They have high populations of mature drones in the colonies that come out of the almonds. They're very beautiful looking when they come out of the almonds, full of nectar, honey, pollen, and mature drones. So they're set up to produce hundreds of thousands of queens. This is just a look at some of the mating locations. They mark them all. They use styrofoam nukes, and they use standard double colonies for drone mothers scattered around the area. This is just a drone shot of some of the mating areas. You see the drone mothers to the right in the middle of that picture, and they're scattered all around. So just an example of the scenery. And one thing I want to note, they go to a lot of effort to maintain our strains. So You'll see this is Y26 where it was mated at, and all the nukes in that area will contain Y26 daughters from one of our breeders. So careful consideration of maintaining the strains. This is some locations in Saskatchewan, our close population mating yard birdies, which you've already seen some data on, and Bainesville, a natural selection yard. Coquets also for a good mating area, more natural vegetation here and bush. So what do? I look at mainly behavioral assays. I reselect the pre-selected stock. Look at temperament, of course. I like bees that are calm on the frame and not running around too much. Low temperature flight. If they fly early in the morning, that's a sign that they will have a longer flight period and be able to gather more resources for the hive. Queen retinue is something I've looked at for a long time, and I'll show you a video on how to look for that. And swarming, superseding, pollen storage, pollen placement, propolis are also important traits, of course brood pattern, and so on. But one thing I'll focus a little more on is drone brood varroa assay, which we do. I used to wash with alcohol or soap, but it's time consuming, and I like to do the drone brood assay quickly on frames with drone brood on them. VSH assays we'll be doing more of. I've started the UBO assay in Canada. I'm going to do it in the US as well as some Harbo scoring. So here's a video showing our varroa assay. I've already done a lot on this comb. I like to do a minimum of 50 and up to 100 and just look at the varroa infestation, and if I find varroa, I use it for an assay. I present the bees with the varroa, and then I watch how they respond to it. Often they jump from the pupa onto a adult worker bee, and sometimes the bees attack them, and so on. So it's something, if you got patience to do, is of interest. This is what I do for retinue. This is something that I do on pretty well all the colonies. Just notice the queen is looking to lay, and the bees are closely attending to her. And you see the retinue form, it's quite obvious. If you don't see that, and the queen is wandering around on the frame, that colony is doomed. Usually she's superseded right away, or the colony fails. The pheromones given off by a good queen show the bees to be very responsive, take care of her, and she lays prolifically. So what we used to do, but now unfortunately the bee-informed partnership is no longer doing this, but it was very good to have this. This is an independent assay scoring for various things: brood, shock broods, temperament, so on, and hygienic behavior by the freeze kill test. So we are scored pretty good. Here's an example of one of our better varroa tolerant or resistant lines, showing very high uncapping and removal scores. And you notice the queen is a nice looking queen with clearly the bees producing good brood and so on. Just to provide some data on what we found in comparing the hybrids produced in California with their performance relative to the stock produced in Saskatchewan. We measured the mites per 100 bees and the honey production over a season, and the bottom line is that there was really no significant difference between the honey production of our queen spruce in Saskatchewan and those in California. If anything, the honey production was slightly better in the hybrids than in our locally produced queens, but the varroa resistance was slightly better in our homegrown queens than in the hybrids. But we need to do this again, and are looking at doing that in the coming year, just to see how things have changed over the years. We trademarked the Saskatraz stock just to maintain the authenticity. So if you purchase the stock, you should be able to trace it back. OHB does a good job of recording and certifying the stock, so we want to make sure that you're getting the queens that they're supposed to be. So if you have any questions, I'd be glad to answer them. We're really grateful to the stock sales to both Canadian and U.S. beekeepers, because that's what funds most of our program. We have had some support from APIS-M in the last few years, and we are grateful for that, as well as the support of the California queen producers in Orland, USDA, and so on. This is just a picture of some of the team members over the years, and you can see who has helped us in our program.